Students, today we are going to study about the prokaryotic DNA replication. Okay, so basically, before going to the replication process, we'll learn about the few basic rules for the DNA replications. So, the DNA replication is a semi-conservative. It is always a start at the origin. We'll consider as the origin of replication, and can be either unidirectional, otherwise a bidirectional. Okay, DNA primers are required, and it is a semi-discontinuous kind of the DNA replication. Always, it is always synthesized from 5' to 3' directions. Okay, you can see here the movement of the otherwise a formation of the replication fork. It is a bidirectional. You can see here, and we have a double helix DNA. Okay, once the DNA, so in the last previous video, we have discussed the different type of enzymes required for the DNA replication, right? So once the double strand DNA it has converted to the single strand, then the newly synthesized DNA will be appeared with the help of the DNA polymerase, and the unwinding it is help of the DNA helicase and single strand binding proteins. They provide the proper template, and the DNA polymerase it polymerizes the new strand formation, and topoisomerase it terminates the DNA replication. We'll discuss in detail. So DNA replication is the basis for the inheritance. So in the this is a cell cycle, right? We have a M phase, and this will be the G1, G0 phase, G1 phase, S phase, and G2 phase. So usually cell cycle we are divided into interphase. This all from like G1 to the G2 phase is considered as a interphase, and M phase we have a mitosis and meiosis as a division, like somatic division as well as a reproductive division. Okay, your replication will be happens in the S phase of the interphase of the cell cycle. Now, the basic mechanism for the DNA replication it is we have a three different steps, like the stages here. One is the initiation, another is the elongation, termination. Okay. So in case of the initiation, the protein bind to the DNA and they form the they'll form the bubble-like structure that is we consider as a replication fork also, and they prepare for the DNA for the complementary base pairings. In case of elongations, it will add the corresponding nucleotide and it will continue the synthesis of the DNA with both the strands. And termination where it is exactly arrest the replication process. So now it's the DNA replication in prokaryotes, the origin. So origin of replication, the ORIC sites will consider as the in case of prokaryotes, you can see in case of E. coli we have 13 repeats and 9 repeats. These are the base pair repeats, right? So they are evolutionary conserved sequences. There exactly where the DNA replication initiation it happens. And you can see, like we have like the three set of the 13 base pairs. Okay, and here we have 9 base pairs. So these are the evolutionary conserved sequences, 9 base pairs. They are rich of the TT, EAT sequences, and 13 base pairs are the GA. These are the sequences. They are also considered as a DNA box, and this 9 base pair will consider as a DNA box. Okay, so here is the initiation point for the DNA replication in case of prokaryote. So this is just another diagram. We have like 13 repeats here and 9 repeats here. From there, your DNA binding protein is will go and recognizes that site, the ORI site set. So from there, it triggers the initiation of the DNA replication process. So we'll go back to the picture here. It is so clear, students. You can see here this our double-stranded DNA. Further to the DNA binding protein A. It will go and wrap the DNA and it will trigger the initiation of the DNA replication. This wrapping of the DNA binding proteins, like the DNA A protein, it initiates the denaturation of the double-stranded DNA. It forms the replication fork and it will initiate the DNA replication. So it will unwind here, then remaining other like proteins, for example, single strand binding proteins and DNA helicase activity, it will start for the initiation, otherwise the elongation of the DNA replication. So you can see here the exactly the how the replication fork it will form. So this is double strand DNA whenever it is separated due to the activity of helicase enzyme and this single strand binding protein, it formed the replication fork. Otherwise, it is also called as a fork replication bubble. Also, this usually the ORI site it is the origin of replication is more of 80 sequences. Okay, instead of the GC sequences. This replication bubble in the form of usually the DNA replication in prokaryote, it is also called as a theta model of DNA replication. The structure it looks like the like theta model of DNA replications. It is also called as a semi-conservative because of a semi-discontinuous phase of replication because the one strand it will replicate in the form of the living strand. Okay, another strand in the form of lagging strand. This strand will be in the synthesized in the form of the fragments. That fragments we consider as a Okazaki fragments. You can say this is a blue strand. It is a new like, template strand up to, and the newly synthesized strand will be always 5' to 3 prime. So here there is no problem with DNA polymerase 3. It will add the nucleotide. So here the synthesis will be in the continuous. But here it has a direction 5' to 3 prime. So DNA polymerase, it will not add the nucleotide 5' to 3 prime. It needs the like the 5' to 3 prime direction, especially OH 3 prime OH is required for the adding the nucleotide. Okay. So in case of initiation, just the it will recognize as the DNA binding protein C, all those things they will recognize as the initiation site. Then your DNA polymerase C, so it will start adding the nucleotide with the help of the primers. So primers they will go and bind to the separated strand. So from there the initiation strand it will add only the few nucleotides. What is primers and are we discussed in the previous video. So you can see here clearly the synthesis always will be the 5' to 3' direction. One strand will be the parent strand will be the 3' to 5'. The newly synthesized strand will be the 5' to 3'. We'll consider as a this like leading strand. So here the parental strand direction will be the 5' to 3'. Here the newly synthesized will be the 3' to 5'. This will be the here also synthesis will happen only in the 5' to 3' direction, but the synthesis will be in the form of patches. And there is fragmentation. It is not a continuous. So we'll consider as a Okazaki fragments. Okazaki fragments are nothing but the Okazaki is a scientist. He named that the fragments. So we'll consider as Okazaki fragments. These are the primers are required, as I said in the beginning, right? The primers are required for the initiation of the DNA replication because DNA polymerase 3 itself it will not add the nucleotide. It needs some kind of the initiation support for the in order to start the nucleotide like complementary base pairs. So once the primers are attached here, what is primers? Primers are the short nucleotide of RNA base pairs. Okay, once primers are attached, then even DNA polymerase it will synthesize the both leading strand as well as the lagging strand. Next, in case of elongation, say elongation, it will continue the synthesis of the lagging strand and leading strand. It will form it will extend to adding the nucleotides, and this will be continued till the termination sequences. Okay. So in case of this is the entire diagram of the both replication of both the DNA synthesis in case of like the leading strand as well as the lagging strand, Okazaki fragments, multiple prime, like in case of the lagging strand, we need the multiple primers because the synthesis will be in the form of fragmentation, so multiple primers are required. Here in case of leading strand, only the one primer is sufficient in during the initiation. So to that itself, it will continue till the end of the synthesis of the leading strand. So once the primer or once the DNA polymerase it recognizes the termination sequences, then primers are removed by the other the kind of the DNA polymerase that is DNA polymerase one. So it will it will remove the primers and the gap will be filled with the help of the DNA ligase enzyme. Okay. So the helicase and primase together will consider as the primosomes here. As we discussed, the single strand binding protein, they'll avoid the unwinding of the separated DNA strands and clamp loaders, it is a part of DNA polymerase. Now we'll discuss in case of the prokaryote. In the beginning, I told it is the replication in the form of circular genome. They have only the single chromosome, and it is a bidirectional. Okay, so how the termination happens because we have bidirectional synthesis, and it is the both side they have a two direction here. So in case of the replication termination, in case of prokaryotes, it will be because of the some ter and tus sequences. We have in one side will be the ter sequences, another side will be the different kind of the task sequences, and here so whenever the replication forks move opposite like they will join with the another direction like clockwise as well as anti-clockwise. When the sequences coincide, then from there it will start terminating with the help of the tar proteins as well as the tar sequences. So these are the tar sequences. For the tar protein it is required, so based on the signal it will stop the DNA synthesis. Okay. Thank you. I hope this video, so now you are clear with the mechanism of DNA replication in case of